Perfect. All right, good morning, everyone. Thank you so much for being here this morning, and thank you so much to the organizers for allowing me to come speak a little bit about the stakeholder work that we've been doing as it relates to cultured meat and seafood safety. We really think this work that we're doing is going to help the industry as a whole and really help us understand the questions that different stakeholders have about these products. So just very quickly, I work for Vireo Advisors. We are a safety and regulatory consultancy. We are mission-driven, so we try to help companies that are making innovative products that are better for the world. I'm lucky enough to mostly work with alternative protein companies. That is my passion. And we try to bring safer technologies to the market. We are made up of a small but flexible team of microbiologists, chemists, risk assessors, but we also work in collaborative work, which is what I'm going to be talking about today. So we work with New Harvest. Hopefully you've heard of them. They're a global nonprofit, and they're trying to build the field of cellular agriculture. They're really good about bringing together stakeholders and transparency. And together we've been working on the Cultured Meat Safety Initiative since about 2019. So, jumping right into it, why is safety research important? So, the number one thing, of course, is that we want to ensure that products are safe. That is our ultimate goal. We want to make sure that there are nothing that goes out into the market that is unsafe. We know that one mishap can affect the whole industry, so we're trying to help everyone be able to adhere to safety standards. The second goal that we have is to increase regulatory efficiency. So we know right now that regulatory agencies are reviewing all the dossiers on a case-by-case basis. These dossiers are massive. They have lots of appendices. They take a pretty long time to review. So our goal is to figure out where we can improve and increase efficiency in the safety assessment. Can we establish inputs that are already safe for use? Can we figure out methods that everyone can use that regulators will accept and find validated for use? We know that regulators all around the world are very under-resourced. They've told us this. We know they're receiving a lot of dossiers right now, so this is kind of our ultimate goal, and that's why we want to work with multiple stakeholders, not just industry, not just regulators, not just academics. We all need to work together to be able to do this. The final thing, and something that I'm going to keep coming back to, is we really need to be able to improve consumer confidence in the safety of these products. We need to have data out in the public domain. We need to make sure that information is readily available for folks to be able to review. And so we want to really make sure, as part of this project, that we can put some data out there. Right now it's pretty scarce, and so we are a little bit worried that the questions that folks are asking aren't being answered in any scientific manner. So we want, consumers to be able to just focus on the flavor, the taste, enjoy the experience, and not be worried that their products are not safe. So we did start back around in 2019. The first phase of this project was primarily focused on industry. So we gathered a lot of folks in industry from about 50 companies at the time, which represented the majority of companies back in 2019. And the main question that we were getting from regulatory scientists and from safety scientists was, we just need to understand what this process even is. So we brought together a number of folks through a series of interviews, one-on-one interviews and workshops, and we came up with a generalized manufacturing plan. This really helped us be able to go through each of the steps and identify any potential hazards that could exist in any of the process. So this paper was published in 2021. I hope you can go look at that if you're interested. It does serve as a nice baseline for companies to be able to identify any potential hazards in your process. However, the next phase, which is the one I want to talk about, is phase 2, which is where we spoke to regulatory scientists. So we know there's a lot of uncertainties, there's a lot of data gaps, and we wanted to hear what they're worried about, what they would like to see more information on. So again, we did another series of one-on-one interviews with over 20 participants from 8 regulatory agencies. And then we held a series of 3 workshops. One was in person at the Agri-Food Week in Singapore last year. And then we did 2 virtual ones to make sure we were accommodating worldwide regulatory agencies. And the goal was to identify the key safety questions and priority areas of research. So this is what I'm going to go through today, so we can let you all know what the concerns may be, where we can be going in future research. As I go through this, I'm going to draw from these interviews and experiences that we had with the regulatory agencies. I will also draw a little bit from my experience with working with companies as we go through regulatory review of dossiers. And also I was part of the FAO, WHO expert consultation on cell-based foods, and so I got a chance to work pretty closely and discuss over 4 days, 4 full days, in depth about all kinds of hazards and issues. So yeah, hopefully we can give you some insights there as to what questions are out there. The other thing I want to point out before I move on too far was that the thing I heard over and over again from experts was that there actually, there aren't many red flags that are major concerns. So that means that there's a belief that a lot of the safety assessments that we already do for conventional foods can be adapted for cultured meat, which is a good sign for all of us. But anything that exists as an uncertainty or a data gap, we need to still get some academic research, some public research out into public domain so that we can be assured that those are not safety concerns. So that's why I'm gonna go through all these in depth today. We have the list of interview and workshop participants, or the ones that were allowed us to disclose their names on the right there. We ran all of the workshops and interviews with Chatham House rules. So that means that we told the participants that we weren't going to attribute any ideas or specific quotes to specific individuals or organizations. So this really allowed folks to be able to speak pretty freely at these discussions. So this did get published. So if you want to go see some of the quotes and read a little more in depth about this project, please do go. This paper got published earlier this year in Foods. And I'm happy to send it to anyone who wants access to it, but it is an open access paper, so it should be free to all. So what were the overall goals or the key findings of phase number 2? Very generically, there still were some concerns that we haven't defined all the hazards associated with novel processes. So we really need to distinguish what's novel, what's already existing, making sure that we're covering all those hazards. There was quite a bit of discussion about what are appropriate methods and approaches we can use. How can we standardize them? How can we harmonize them? Maybe we need to do some modification from what already exists to make things a little more efficient. And something that I will keep coming back to over and over again is this idea of data sharing and publicly available data. As I mentioned, regulators are getting swamped right now, not only from dossiers, but also from public questions. So they're getting calls directly from the public asking about the safety of these products. And so if we are able to provide some sort of scientific evidence for these, that would really make their jobs a lot easier and they'd be much happier with us. So this is from the paper. I'm not gonna go through in depth, but because I will be talking about all these topics in this presentation. But generally, we need to understand the process, understand the product, and then of course develop methods. What I want to really highlight is that we're hoping these will feed into databases, help develop standard safety testing methods, and the most important thing is like creating an evidence-based path to safety assessment. So what are the research priorities? The first were involved about understanding the production process, characterizing the inputs, genomic and metabolomic stability of cells, lots of discussion about this, and some open questions still about any novel toxins or allergens. So first, concerning the production process. One of the things that was discussed, and I have heard this multiple times at different conferences and roundtables, is it would be really great to be able to understand where there are areas of convergence across the majority of processes. So this may include things like the equipment, the inputs, and what I mean by this is if we understand what's common to most processes, we can then develop either templates for safety, we can do overall assessments or general assessments of these components and put those into public domain. And so then you companies can kind of point to that rather than having to do your own complete risk assessment and submit that in a dossier at this point. So currently, everyone is doing their own risk assessment on everything, creating their own safety plans. It's a lot of work, and it also is a lot for regulators to review, and it's sometimes repetitive. So it'd be really nice to be able to kind of narrow down on what these things are. So things like bioreactors, how can we safely use them, what hazards can be introduced. And then one idea that has come up so many times that I've heard is the basal media. So we know that the basal media is made up of lots of components that are very common in conventional foods, amino acids, vitamins, minerals, so forth. So if we could just have a database of all these substances, their safe levels, safety data that demonstrates that these are safe, then it'd be much easier for everyone who maybe is an R&D or looking to use different components in their media to be able to select these and know that they're safe for use and see that there's safety data already to support the use of these. Something we still heard from regulatory scientists was that they still don't quite understand how antibiotics are going to be used and at what stage, and if so, do we need to develop guidance for that? There have been some questions about whether or not we have definitively identified all the potential microbiological or chemical contaminants that can come as part of the manufacturing process or as part of the inputs. So having a research project to be able to, identify everything would be wonderful. There were questions about whether or not recycled media, so if you're recycling media, does that change the composition of the media as it goes back into the process, and will that affect how your cells grow and the composition of those cells? Some questions that we heard a few times about that. And then we know that good manufacturing processes and HACCP plans have to be developed as part of your safety plan and running of your facilities. So it'd be really nice to be able to develop some templates that would be appropriate for cultured meat processes. The next big topic, of course, are how to do the safety assessment of inputs. We know there's a lot of different things that go into the product. So it would be really nice to have more standardized guidelines to assess the safety of inputs. How do we actually go about that? Do we measure them? Can we do other indirect methods where we can kind of do worst case estimates of how much of the input would be in the final product? One of the very, very hot topics right now, and I know this came up at the Agri-Food Week Regulator Roundtable just a few days ago, is the topic of growth factors and bioactive molecules. So we know this can be, there's concern about these because these bioactive molecules theoretically have the potential to be active in our own bodies as well. And so the Singapore Food Agency has put out some nice recommendations and requirements to assess these, but there still is a lack of data out in the public domain about whether or not these bioactive molecules remain bioactive in final product. Are they going to be present in cultured meat? If they are present, will they be inactivated when you cook the product? Will they be inactivated when we digest the product? So far, at least for the first generation of growth factors that are similar to those in food, we do know they're quite unstable, but we do know that coming down the pipeline, we're all trying to use more stable growth factors, more bioactive growth factors, and so having a set way to assess these would be very beneficial. There were some questions about whether or not some substances are going to bioaccumulate in a product, so things like heavy metals. And something that would be really helpful for everyone is this idea of what is a food-grade substance. We don't have standards for that right now. So having some specifications for various inputs would be really helpful. The codex or having international organizations involved in this would be really good. And then in one of the kind of interesting questions that we heard a couple times from non-cultured meat scientists, they said, well, we know that there are some substances commonly used in cell culture that maybe some groups are still using because that's just common to growing cells. So things like phenolphthalein, which is a pH indicator, not necessarily have any sort of direct use for food, but could be used in cell culture. How do we assess these? How do we make sure these are not being used? Are they safe for use in food? So just thinking through those types of substances as well would be very good. All right, genomic and metabolomic stability of cells. So this idea of whether or not the cells we're using when we're doing prolonged passages, is there a possibility for genetic drift? Are these cells stable? How do we demonstrate they're stable? There's no easy or cheap method really to do this in a very holistic way right now. So the questions are, what parameters should be tracked and evaluated? So we know from the 2 dossiers that have already been reviewed by the FDA, we know from some papers that we've seen in peer-reviewed literature like that by Believer Meats that there are some approaches, but there's definitely no harmonized way to do this yet. So we know that some groups are looking at morphology and growth characteristics to make sure that they're consistent. There are a number of omics, genomics, proteomics, transcriptomics approaches where you can compare your cell lines to conventional and to look to see whether there are any differences, but there's no established way to figure out what are the key differences, what are the thresholds, what kind of drift are we allowed to see. We've seen some karyotyping being done, so chromosomal stability. One of the challenges is the question for genetic stability that we often get is, are there any novel toxins or allergens being produced when your cells are mutating as you're passing them? And obviously this is an extremely difficult question to look for things that you don't know what you're looking for. So one of the academic scientists that was part of these groups did say that we need to do maybe some sort of research project that has a hypothesis-driven analysis, so doing the thought exercise of thinking about what is possible to even be produced in these cell cultures, what could be dangerous, and then do some targeted analysis to look for those. So yeah, that could be a very large research project. Very quickly on novel toxins and allergens, obviously some cell lines have the potential to produce some allergens and toxins and toxic metabolites. Usually this is pretty self-regulating in the cell lines. They don't survive if they're producing those, but it was still an open question. Then we got to a pretty large discussion about which testing methods are appropriate. So the first was this comparative assessment. So this is currently how a lot of assessments are done. And currently we basically compare the cultured meat cell lines to something that may be a conventional product that we already eat. There were some questions from the regulators that said, what if we're using a cell line or some sort of species that you can't easily get some tissue from the conventional animal, so something like endangered species. How do we establish a conventional comparator in those cases? It's really hard to get those kind of data. I will say that regulatory scientists did acknowledge that differences are expected and not necessarily a food safety issue. So things like protein content and amino acid ratios. And of course, we know we are introducing some novel things like scaffold. So that's not a complete surprise. And they also acknowledge that there already is a really wide range of natural variation in conventional meat. So we would also expect there to be some sort of variation in cultured meat. But the question is, what kind of variation do we expect? We don't really have great data to be able to compare to right now. A lot of the compositional databases are like for conventional meats they're not ranges, they're just like one number of one person who's ever measured that meat. So yeah, really not great to compare against right now. There was a question about nutritional specifications. Do we need to develop those? This was in the context of long-term consumption. So in the future, if these proteins do start to substitute or replace conventional meat products, do we need to be worried about different vitamin levels, different amino acid levels, protein quality, that kind of thing? So still some open questions there. And then the actual approach to testing the inputs, there were some ideas that were thrown out there. The first was just about, we had some conventional food toxicologists in the room. So they were wondering whether or not we should be testing the final product, so a whole product, versus individual substances that go into the product. And if those are all safe, then we can assume that the final product is safe. Up to this point, we have been doing more or less individual substances. Conventionally, we would do a 90-day subchronic animal dietary feeding study. That's obviously something we do not want to do, but it's also incredibly challenging. There's almost no way we could do that with meat. It would be, rats don't want that for 90 days straight at high levels. So yeah, that's one of the questions that was out there. There was some agreement that for substances that are already part of conventional animals or part of cells. We could do comparative assessment again, so looking, comparing conventional versus cultured meat. There was some ideas about how to assess exogenous substances, especially those substances that have never been part of the food supply or have been added to the culture that we wouldn't normally expect to be in meat. Some things like small molecules. One idea is this use of this threshold of toxicological concern as a TTC approach. So rather than having to do a full suite of testing for that particular substance, this sets a threshold for which different substances can be used. So we look at the structure of the molecule, we then look at other similar substances and we kind of categorize how hazardous that substance may be, and then we say, okay, you're class 2, you can't be above a certain threshold. If you are below that threshold in the final product, then you're not of concern. So rather than having to go through the full suite of testing, we can kind of use that approach. Allergenicity is already really challenging to do in even conventional products. We use a weight of evidence approach where we may first start looking at the sequences of the proteins, compare it to known allergens, go through in vitro testing. If there's concern, you can go all the way up to animal or in vivo or human analyses. So this is something that is still a bit of an open question if you have any sort of novel proteins that may be allergenic. And then very basically, so there were questions about whether or not just our simple monitoring, sampling frequency, what are the standards for those once you start doing commercial production? So for example, for microbiological contamination, do we use the same standards that we use for conventional meat, or should we come up with some new ones? All right, so I'm gonna finish off the research priorities with this idea of databases. This came up in almost every single subgroup that we had during the workshops. So I think that it is something that we really want to delve into in our next phase of work. And that is basically developing some databases for cultured meat products. So currently some databases definitely exist for a number of other factors, but of course at this point nothing includes cultured meat data. And so we would like to be able to work together with the community and collaborate to be able to develop databases that may be useful for us as a whole. So maybe inputs, like looking at all the basal media components, maybe some unique components that are present in a lot of different culture, figuring out their safety thresholds, figuring out whether or not they're present in final products, something that the community could use as well as regulatory scientists. And really, I just want to emphasize and bring home again that the idea of all this research is to identify all credible risks. So that's obvious to all of us. But also, we need to rule out non-risks. So when we have questions, for example, about the carcinogenicity of a cell line, we can have a dialogue about that, but if there's no information in the public domain or very little information in the public domain, it's very hard to make a scientific argument about it. So we did identify a number of research gaps. Again, I want to emphasize that many of these experts did believe that the products are not that different than conventional, that we can use methods that already exist. But again, because there's a lack of information, it can feel like there's a real lack of transparency, and so that can really affect us all. So just want to put out a pitch out there. We are moving into phase 3. We're starting to create some case studies and some smaller collaborative groups to be able to tackle some of these issues. Our vision is to be able to bring in industry, regulators, academia, folks we've already spoken to do realistic academic research, so using actual products, doing things that make sense. We know that academic researchers are really keen to do this work, but they don't necessarily want to go off and do things that aren't realistic, because that's not really going to help us in the long run. So we are asking for folks who are interested in this type of work to please come speak to me, to contact us at Vireo Advisors or at New Harvest to be involved so we can be talking about phase 3 of this work next year. We would really like to develop some public-private partnerships, work with government to also see what they need as well. So with that, I'd like to thank you. This type of work can't be done without donors. So New Harvest received a lot of funding in order to be able to run this project. Also, thank you, of course, to the New Harvest team, who are amazing collaborators to work with. The Singapore Food Agency were our co-hosts of the workshop. They donated their space to be able to hold the workshop. And yes, please do contact me. I will be here for the rest of the afternoon. And I have one more slide that I hope got added, because, okay, so this is completely unsolicited, but there were these books in the speaker room that they said we could take. And so Anita and Alex have written this book. And this morning in my hotel room, this is my mom. She's actually here with me on this vacation. She's not a scientist. And I come out into her hotel room and I hear her going like she's making these very excited sounds. And she's reading through this children's book. And I was like, Mom, what are you doing? She's like, I'm reading this book. And she turns to me and she's like, I now understand what you do. And I've been talking about it for 4 years. And so I think this just really highlights how important it is to get information out into public domain, no matter what kind of information it is. It made me really, really happy to see that. And so yes, thank you so much. Yeah, that's all I have to say for now. Thank you.
Thank you so much, and thank you for the shout out as well. Does anybody have any questions? We see one question in the middle there, but I'll run the mic to you.
Thank you for your presentation. What is, in your opinion, the advantage on measuring on the ingredient level rather than the final product? Because I can only see advantages in the end, this is what we deliver as a product, so that's where all the tests should be run. But I hear this argument quite often, so yeah, I was interested if you could elaborate on that.
So the question is, what would be the disadvantage of not measuring it in the final product?
No. Why would be more advantage, or what would be the argument for measuring at the ingredient level rather than at the final product?
Yeah. So as we know, or most of us know, there's a lot of substances that go into cultured meat, or there can be, and more complex processes that maybe include, more types of cells or more types of, especially as we kind of get down the road. It may not make sense to measure every single one, especially if we're already pretty assured of the safety kind of up front. And so if you can do a worst case, for example, measurement or calculation rather, of what you would expect, if there's 100% that ends up in the final product, you may be able to make a pretty convincing argument of safety before you even get to the final product. So you see, even if there's 100% of that component ends up in the final product, maybe you can rule out the need to measure that, because there's already a lot of things that we have to measure, and anything we can do to make that assessment a little faster and more efficient, still making sure it's safe, we're all for that.
So I'm hoping this is a softball more than anything. But you list everything out in terms of submission in a dossier. How do you apply that? Or when do you apply that? When is the appropriate time to approach either someone like yourself or start to think about this? Is it conception, or is it proof of concept? Yeah. Or when do you start thinking about this process?
Thanks, Andrew. So the question is, when do you start thinking about your dossier and your regulatory submissions? I mean, we always encourage folks to be doing it as early as possible during the conception, R&D stage. There are some substances that you can red flag right away. We know that there are some things that regulators will be concerned about or ask for a lot extra safety data, or could be a problem down the road. So that's kind of a high-level assessment in the beginning is always helpful. Same thing for creating manufacturing facilities or buying equipment. All that stuff is so good to do kind of early on. Maybe it's not really helpful for you if you have a product that's amazing, but then you find out you need to remove one of the most important parts of it because it is going to cause safety issues. So as early as possible. Obviously, there are some things like measuring residues and stuff like that. Once you kind of have a close to commercial process or something that you're pretty confident in, then you kind of do different steps at that stage, but as early as possible is always what we encourage.
We have time for one more question. I see some consolidation here.
high respect in this group for everyone that's here and what we're doing together. We've been optimizing stem cell media for quite a while, since around 2019, and there are some components you use in a fully chemically defined stem cell culture media. Some of those small molecules, like a fibroblast growth factor, has a polyhistidine tag on it, which we use a nickel column to bind the his tag to, and then before we elute it. So, you mentioned some of these red flag components. There's another one, sodium selenate is toxic, and 10 micrograms for a child could kill them, but it's important in a chemically defined media. So we're now asking ourselves, should we be, removing these ingredients before we get to a final stage product? Do you have any experience in particular, because growth factors are the first thing we have in the market, if we're selling it to cultured meat companies, should we be thinking about a different way to make growth factors other than standard recombinant manufacturing technique with nickel columns and his tags?
So I'm not sure at this point if there's a need to completely change what we already understand, but you bring up a great point in kind of sometimes we don't think about what it takes to make that substance and what could be introduced. So being able to go back and say, like, let's test for the amount of any sort of, nickel or anything that's going to be in that product. There are safe levels for a lot of these substances in food that already exist in conventional foods. So as long as we can kind of address those and test for them, I'm not sure if we need to completely reinvent the wheel. We have heard that for growth factors that have tags that make them more stable, we do get questions about allergenicity, stability, does it change those? So yeah, that may involve a little more assessment for those types of things. So that's kind of why we like to do it, the assessment kind of up front. A lot of regulatory agencies are pretty open to going to talk to them early on to see what kind of concerns they have. So before you really get too far down the process, we can go talk to them and see what the concerns are and how many extra tests they may require.